Okay.
And I'm going to do the thing where I say, if you have internet bandwidth, please turn on your video camera. It is not fun giving talks to black boxes full of names. Part of why we do this is to build community, and it's really hard to build community when it's just a black box. Seeing people's smiling faces is an important part. Thank you, Dominica, for being a leading fellow today with their video on.
All right, I'm assuming a couple more people will continue to join. I think we tend to have some stragglers, but I will maybe start us off. Oh, Eliza keeps entering the waiting room. I've admitted now the third Eliza Gerstein. I don't know what's
happening. This is very troubling. Okay, who's logged in as me? Seriously.
Anyways, we'll see how this goes. I will continue to try to admit people while introducing our speaker for today, Dr. Zoe Hilbert. So Zoe earned her PhD in biology at MIT. As a grad student, she worked in the lab of Dr. Dennis Kim. She studied patterns of sex and strain-specific neuronal gene expression in the nematode C. elegans. And for her postdoc, Zoe sort of shifted course a little bit, moved to the University of Utah, where she worked in the lab of Dr. Nels Elde, where she transitioned to spending her days thinking about pathogenic fungi, which of course is why she has landed herself here today, and the evolutionary conflicts and conservations that play out in host fungal interfaces. And so this is actually something I feel like we haven't had a ton of speakers on in our seminar series. So I'm very excited to welcome Zoe, who's just very recently started her independent research lab at Boston College, where her group combines evolutionary genetics and experimental evolution approaches to try to understand the mechanisms of fungal adaptation to new environments and host species, and identify how fungi have shaped the evolution of the mammalian immune system. So we're delighted to have Zoe here today, and we'll let her take it away.
Great. Let me share. And hopefully you can see the normal screen and not my notes. Is that true?
Yep, we're seeing your normal. Yep.
Okay, perfect. All right. So thank you, Rebecca, so much for the invitation and for the introduction. It's really exciting to be here today to talk to fellow lovers of evolution and pathogenic fungi. And so like Rebecca mentioned, my lab just opened at Boston College in January of this year. And in my lab, we're really interested in trying to understand how the interactions between fungi and diverse species and environments really impacts the evolution and adaptation of these fungi. Now, we use many different approaches to answer these questions, but the main one that I'm going to focus on today is using experimental evolution methods. And to us, these sorts of experimental evolution methods have a lot of really important advantages, and I'll just give you two of my favorites. So one is that we find that these approaches can reveal genes and pathways that are involved in adaptive processes across wide-ranging conditions, and that this often shows us the unique methods that fungal species in particular use to adapt to these different environments and give us a really nuanced understanding of the biology of these organisms. The second reason that I particularly love experimental evolution approaches is really it allows us this opportunity to watch evolution unfold before our eyes. And so not only can we do these sort of mechanistic studies and get down to an understanding of the genes that are important for adaptation, but we can actually learn something about the evolutionary process itself, which I find really quite amazing. And so in my lab, we use these sort of experimental evolution to ask questions about how all sorts of different environmental factors, but also interactions with all sorts of different species influence the evolution of these fungi. But for today, I'm really just going to focus on two stories. So I'll start first by telling you about work that I started as a postdoc looking at the role of different host species in influencing fungal evolution. And then at the end, I'll tell you a little bit about a brand new story where we pivoted to using similar experimental evolution methods to ask questions about how our changing climate might be impacting fungal evolution in interesting and sometimes scary ways. Okay, so starting first on this host side, this is really work that I started in my postdoc in Nils Aldi's lab at the University of Utah, but which is also carried with me to my new lab, and we're still following up on a lot of this stuff here now. And so really the genesis of this project came from a fascination that I developed when I started my postdoc with the question of how fungi actually became pathogens in the first place, and I should say pathogens of mammals in particular. And so when we think about where we find most of the fungal pathogens that infect us humans, their primary location that they prefer to live is out in the environment. So in the soil, on trees, associated with bird guano and other places that look very different from inside of a mammalian host. And so how do they actually become successful across these many different environments? And one of the hypotheses that gets put forward all of the time to explain how pathogenicity evolved in these fungi is that they're actually trained through their interactions with environmental host species that they encounter. So these include species like amoeba and nematodes, and that through their interactions with these environmental predators, these fungi kind of learn the tricks of the trade of how to succeed inside of a host cell. And that leads to these adapted populations that are then poised to be able to succeed within the context of a mammalian host. Of course, we know that evolution doesn't actually stop once the fungi get into the mammalian host. And so I was also really curious about how repeated interactions between these fungal cells and host immune cells like macrophages might also drive these cycles of adaptation with really important implications for disease progression and outcome. And so what I wanted to try to do was to ask whether we could provoke this sort of evolution of a fungal pathogen in a lab. So could we build a system where we could actually drive this evolution and use it to ask questions about how these diverse host species actually influence the evolutionary trajectory of fungi? And so the system that I dreamed up looks something like this, where I imagine we could take environmental strains of fungal pathogens, really trying to understand how these strains go from the environment into the clinic. We could expose them to host cells from different species and allow these to interact over defined periods of time. At the end of this, we could selectively lyse those host cells, collecting only the fungi. We can freeze down aliquots, which creates what we like to call our fossil record and facilitates those evolutionary studies that I love so much downstream, and I'll come back to this later on. And then we take the remaining fungal population and repeat this process over and over again and see what we get out. Now, before I show you any of the data from this system, I first want to introduce you to the players that I'll be talking about today. And so we'll start first with these host cells. So as I've already alluded to, all of the work that I'll be showing you was done in parallel in both an amoeba model as well as in a mouse macrophage-like cell line. You can see both of these in these images here. We'll start first on the left with the particular amoeba that we work with, which is a species called Acanthamoeba castellanii. And in this video that I'll start in a minute here, you're going to see these amoeba crawling around and interacting with these fungi. These are yeast in their culture media. And so you can see they move around quite a bit. They interact with the fungi, and if you get really lucky, you might see one actually phagocytose one of these yeast cells. And the particular strain that you're seeing in this image that we work with in the lab was actually isolated as a contaminant from a culture of wild yeast. It's also really abundant in the environment. If you just go out and sample some soil, probably anywhere, you will find these crawling around in the dirt. And so we think they are actually really an abundant and ecologically relevant predator of many different types of fungi. On the right, you're seeing an image of the J774A cell line. This is a mouse macrophage-like cell line, and you'll see a video of these as well interacting with those same yeast in their culture media. They're much less exciting to watch. They don't crawl around. They don't have as much charisma, in my opinion, but they do phagocytose quite a bit of yeast out of their culture media, and this is going to serve as the macrophage model for all of the work I'll show you today. Okay, so those are our host cells. What about our pathogen? And if you have a keen eye, you might have already detected that the fungal pathogen that we decided to focus on for this work is the human fungal pathogen Cryptococcus neoformans. This is probably familiar to some of you in this audience, but just to remind you, this is an encapsulated yeast species, as you can see in this image over here. So it produces this really distinctive polysaccharide capsule to protect itself against all sorts of different insults, including host immune cells. And it's a member of this basidiomycete group of fungi. So it's a yeast and looks very similar to some of our other favorite yeasts, which you might find up here, but it's actually separated by hundreds of millions of years from some of those other commonly studied yeast species. Now, like many other fungi, Cryptococcus is in particular a really big problem for HIV and AIDS patients. So it currently accounts for about 20% of all AIDS-related mortality every year. And it does this primarily through its tropism for the brain, where it causes a particularly fatal cryptococcal meningitis. And this dissemination to the brain and to other sites within the body is thought to happen in part through its ability to colonize macrophages and use them for transport throughout the body. And so this sort of further motivates our interest in studying adaptation to this particular cell type. Now, there are many reasons I could go on and on about why crypto is so great and why it is particularly a great model for studying fungal evolution and experimental evolution specifically, but just to highlight a few for you here. So it is a haploid yeast, so that makes genetics pretty easy in this species. We can also grow up large quantities of these relatively quickly, which makes these evolutionary studies really quite feasible. It's also genetically tractable, which facilitates the mechanistic work that we do downstream of our experimental evolution. And then really importantly, given our interest in trying to understand how these fungi go from the environment into the clinic, there's been quite a lot of community interest in trying to understand the evolution of virulence in this species. And so to this end, there's been a lot of sampling of Cryptococcus strains, not just from clinical samples, but also from the environment. And so what you're seeing on this phylogeny over here is more than 300 different Cryptococcus strains that have been sampled from all sorts of different locations. And I don't expect you to take a whole lot away from this tree other than to appreciate that these strains sort of divide into 4 major lineages with different levels of genetic variation across these lineages, as indicated by these different branch links that you're seeing here. And what I think is particularly interesting is that most of the environmental isolates that have been found so far cluster into these V and B lineages where we see most of that genetic variation. The clinical isolates have far less of this. And so I think that's one of the things that my lab is particularly interested in trying to understand through lots of different methods. Now, this is also an incredible resource for doing these sorts of evolution studies and for identifying strain variation, which can feed into our experimental evolution setup. And so what I did was I identified 34 strains across this phylogeny coming from all of the 4 major lineages. These are highly enriched for environmental isolates, but there are a few clinical ones shown here with the black dots that we've thrown in as sort of interesting comparison points. And so we were very nicely sent all of these strains by John Perfect's lab at Duke. And when I got them into the lab, the very first thing that I wanted to do was to see if there's any variation in how these strains are actually able to replicate in our 2 host cell model. And so before I show you the data, I'm just going to quickly walk you through how I do these experiments, since it will form the basis for a lot of the data that you'll see in the slides to come. And so to test how these strains are able to replicate in our different host cell types, what we do is we take a strain of interest indicated as these round orange cells here. We put these Cryptococcus cells into culture dishes, either with macrophages or with amoeba, and we allow the Cryptococcus to be phagocytosed over the course of 1 hour. When that 1 hour is up, we remove any of the yeast that remains extracellular. So anything that didn't get phagocytosed gets thrown away. And we allow everything to grow together for an additional 24 hours, at which point we lyse our host cells, collect our fungi, and plate so that we can count CFUs. Importantly, we also plate a series of controls after this initial 1 hour phagocytosis step. This allows us to assess differences in phagocytosis across strains, but also lets us get a sense of how much replication is actually occurring in these host cells over the course of that 24-hour period. And so what does the data look like for these strains for host replication? So starting first with our macrophages. So I'm showing you here just a subset of the strains that we've looked at. Each strain is given its own color here. And I've plotted this data such that on the y-axis, I'm showing you the log2 of the CFU fold change over that 24-hour growth after the phagocytosis step. So essentially, you can think of the height of the bar as indicating how much growth each strain has inside of macrophages. So the higher the bar, the better it is at growing in the macrophage. And so hopefully what you can appreciate is that all of these strains are able to replicate in macrophages. All of the bars are above the x-axis, and there is some amount of strain variation. But on the whole, most Cryptococcus strains that we've looked at are actually surprisingly good at replicating inside of these host cells. We can look at the data for the amoeba plotted in the same way. So that's now shown here on the bottom, and immediately you will appreciate that this graph looks really different, even though it's plotted identically. So this is the same subset of strains. The y-axis again is showing you here the log2 of the CFU fold change inside amoeba this time. And what you can appreciate is that all of these bars are either falling at the x-axis or below it. And so what this indicates is that the amoeba are actually really quite good at killing our Cryptococcus cells. And this has been kind of universally true across most of the strains that we've looked at, with a few kind of important exceptions. So one of them you're seeing here, this NRHC 5004 strain, which does seem to be able to replicate to some extent in our amoeba. It also does much better in macrophages. And so we have a few examples like this that we're currently using other methods to try to understand how they're so generally good at growing inside of host cells. But instead of continuing to take you through all of our phenotypic variation that we've uncovered, I'm instead going to focus on just one strain for the rest of this part of my talk, which is the strain here shown in teal called FTC 555-1. This is an environmental isolate that came from a mopane tree in Botswana. And we selected it for our experimental evolution approach because it doesn't seem to grow very well in either the amoeba, and also comparatively grows worse than most strains in our macrophages. So we reasoned that we had some room to improve the ability of this strain to actually grow inside of these host cells. And so what we did was we took this FTC 555-1 strain, we put it into our serial passaging system, which looks really similar to what I showed you on previous slides. And we passaged it through our host cells for a total of 12 rounds of passaging. We independently evolved 6 populations. 3 of these were passaged through amoeba, and 3 were passaged through macrophages. So I probably wouldn't have set all of this up if we didn't find something interesting, but what did we actually see as a result of this passaging? I'll start first with our amoeba passage strains. So the first question we wanted to ask was, we just passaged these through amoeba for a number of generations. Do they actually get better at growing inside of these amoeba host cells? And so that's what you're seeing in this plot here on the left. On the far left in teal, I'm reminding you of our starting parental phenotype. So this is where we started. The strain was killed by the amoeba. And in these 3 different shades of green indicated by the 3 A strain names, what you can see is the phenotypes that we recover after this experimental evolution. And I think this data really beautifully illustrates the power of this system to reveal different evolutionary trajectories in our different replicate populations. So what we can see is that in our A1 strain shown in the darkest green, we now have this much more robust ability to replicate inside of the amoeba. Our A3 strain remains unchanged, and the A2 strain is somewhere in the middle. Now, I started all of this by telling you that I was really fascinated by the idea that these amoeba might be able to select for traits that are sort of generally useful, not just in amoeba cells, but also in mammalian host cells as well. And so we also wanted to ask whether these amoeba adapted strains are now better able to replicate in our macrophage system. And so that's what you're seeing here on the right. So again, our starting phenotype is shown in teal on the left-hand side of the graph. And our 3 different amoeba passage populations are in these shades of green. And what you can hopefully appreciate is that there's really no change in their ability to replicate in macrophages at the end of this experiment. So even though we're seeing these changes in amoeba growth, that doesn't seem to generalize to other types of host cells. We can also look at our macrophage passage populations in the same way. So again, first starting by looking at how well these macrophage passage strains are able to now replicate in the same host cell they were passaged through. And so that's what you're seeing here. So in shades of blue are our 3 different macrophage passage lines. And we were really excited to see that in 2 out of these 3 lines, we see this really robust enhancement of their ability now to grow in the macrophage host. We asked the same question as to whether these strains are now better able to replicate in amoeba. And again, the answer is no. So we see really no change in these macrophage adapted populations in their ability to replicate in amoeba cells, or if anything, this M1 strain actually seems to have gotten a bit worse. And so I think what that means is that in this particular experiment, what we've done is really efficiently host adapted these strains to the specific hosts that they were passaged through, but this hasn't selected for any sort of general features that confer pathogenicity across all different hosts. This, of course, is still really interesting and I think allows us to ask questions about how this host-specific adaptation occurs. And so for the rest of this part of the talk, I'm just going to focus on our work to characterize one of these strains, this macrophage adapted strain shown here called M1, which had really the most dramatic change in its ability to replicate in macrophages at the end of the experiment. And so the first thing that we did was we wanted to know what had actually changed in the genome of this strain as a result of this passaging. And so I selected both single colonies as well as a pooled population culture and sent them all out for whole genome sequencing. And when I got the data back, I was really pretty surprised to find that there was only a single mutation that differentiated this strain in all of the both pooled population cultures and colonies from the parental strain. This mutation is a single missense mutation that falls into a gene in Cryptococcus called CAC1, and CAC1 encodes the adenylyl cyclase, which is required for the production of cyclic AMP and kicks off this cyclic AMP signaling cascade, which really regulates basically all of cryptococcal biology, including many of the features that provide this species with its virulence. So it's important for regulating virulence factors like capsule, melanin production. It also allows cryptococcal cells to change their cell size, which is an important part of dissemination and infection, and many other sort of more fundamental aspects of the biology of this species. And so the fact that this was really the only candidate mutation that I had when paired with the central role of this gene in regulating many aspects of cryptococcal biology, that really suggested that this was probably the causative mutation, but we of course wanted to confirm this experimentally as well. And so what we did was we took our starting FTC 5551 parental strain and we introduced just this single nucleotide change at the endogenous locus using CRISPR. And what we see when we do this is that this is sufficient to confer this increase in replication in macrophages up to the level of what we observe in our evolved M1 isolate. We can also do the reciprocal swap where we take our M1 evolved strain and we put back in the ancestral allele of CAC1. And when we do this, this abolishes the ability of these strains to replicate now as well in macrophages, really telling us that just this single nucleotide change that causes this missense mutation is both necessary and sufficient to drive this increased macrophage replication phenotype. So this was super exciting. But of course, I told you earlier that one of the things that I love so much about these experimental evolution approaches is our ability to watch evolution in real time. And so I really wanted to be able to do that with this particular experiment and to understand when this mutation arose and what it looked like in the population over time. And so to do this, I went back to that fossil record that we froze down over the course of our passaging. And I thawed out samples from every passage and used these in order to track the dynamics of this CAC1 mutation over the course of every passage of this experiment. And so the results from that are what you're seeing here, where on the y-axis, I've plotted the allele frequency of that evolved CAC1 allele over the course of our 12 passages. And so what we can see is that this mutation arose de novo in the fourth passage, so relatively early on in the experiment, and it existed at relatively low frequency, but then very quickly swept through the population. So within about 2 populations, it was over 90%. It was contained within over 90% of our cells, really suggesting that this mutation confers a strong fitness advantage for growth in macrophages. And I'll say in data that I'm not going to show you today, this actually really nicely matches our experimental data looking at these mixed populations from over the course of our experiment in actual macrophage cells as well. Okay, so we know the causative mutation and we can also see how it swept through the population. We can track dynamics over time. This is really cool. But how does this single amino acid change actually cause such a huge change in phenotype and such a large fitness advantage? And so I'm going to disappoint you all a little bit and tell you that I'm not actually going to get to mechanism today. We do know a lot about how this particular mutation is affecting downstream virulence-related phenotypes, and I'm happy to talk more about that during the questions, or you can also see all of this data in the paper, which was published last fall. But for now, I'm just going to go back to some evolutionary approaches and a little bit of structural modeling to talk a little more about the allele we recovered and what I think it tells us about the power of these experimental evolution systems. Okay, so one way of getting at what the functional significance of what this mutation might be is by using protein alignments to look at conservation of this particular amino acid site. And so what we did was we pulled a number of orthologs of the adenylyl cyclases from many different fungal species, including both pathogenic ones, but also a number of non-pathogenic species. And largely, we don't see high conservation in the sequence across these species. But if we look at the site of our mutation that we recovered, this arginine 1227, we see that most fungi do actually have a conserved arginine here. And in the species that don't have an arginine, we don't see a mutation as disruptive as the proline that we recovered in our evolved population. Even more intriguing, when we look at sequence alignments for all of the more than 500 sequence strains of Cryptococcus neoformans at this point, we see that this arginine is perfectly conserved in every single one of those strains. And so I think that really suggests that this particular amino acid is probably playing some important functional role that contributes to the successful function of CAC1. And so to me, that really suggested that this is probably going to fall in some obviously important functional part of the protein. And so to look at this structurally, we modeled CAC1 using AlphaFold. And so that's what you're seeing here on the right. I've highlighted the two kind of domains that we know the most about in different colors here. So the catalytic domain is shown in this lighter purple that you can see sort of across the protein. And then in blue, you can see this characteristic leucine-rich repeat. This is a feature of all fungal adenylyl cyclases. And we think that it's probably involved in coordinating interactions with regulatory protein partners, but we don't know a whole lot about what it does. So of course, I thought this mutation would likely fall within this catalytic domain. It's clearly conserved in all of the Cryptococcus neoformans and many fungal species that we looked at. But when we actually located the position of this mutation, it fell right here. So it's on the concave surface of this leucine-rich repeat region. We know absolutely nothing about what this particular site could possibly be doing. And so I find this really kind of amazing. I would actually challenge anybody in this audience to look at the structure of this protein and pick out this particular amino acid site as being important for its function. But I think this really highlights the beauty of these in vitro evolution systems to reveal new biology, even in these well-studied signaling pathways that we think that we know so much about already. Okay, so I'm not going to go any further on mechanism here, but again, happy to talk more about that later. But I just want to close with one last experiment, which is probably something that many of you are wondering about already, which is I did all of this experimental evolution in vitro in the lab in cultured cell lines, but what do these evolved populations actually look like in the context of an animal infection? And so we did the really simple experiment of just taking our parental strain and our evolved M1 strain that grows really well in macrophages in vitro and infecting mice with these and doing a simple survival analysis. And so you're seeing the results from that here. You don't need me to tell you that there is a big difference in survival between animals infected with these two strains, but the results were actually quite surprising to me. So in teal here, you're seeing our parental strain. So animals infected with this strain do develop symptomatic disease and eventually succumb to this, although I will say that this is quite a bit slower than what we see for most control strains. So this is not a particularly virulent strain to start with. But in contrast, if we infect animals with our M1 strain, and again, this is the strain that grows so well in macrophages, what we see is that these animals are entirely fine as far out as we've taken these experiments. And so this was really pretty curious to me and a bit surprising. But one of the things that I was sort of immediately wondering about was whether the M1 strain was simply cleared very early on in infection, and that's why the animals are surviving so long. And so what we did was we just took both the lungs and the brains of the animals at the endpoint of this particular experiment, and we plated them to see if we could recover any Cryptococcus from these organs. And so that's what you're seeing here on the right. And what you can see is that we do, in fact, recover crypto from both the lungs as well as the brains of these animals. So not only are they not clearing the infection, we're actually seeing some amount of dissemination with which we normally associate with kind of infection endpoint. And so I think this is really pretty fascinating and suggests that this strain is establishing a persistent infection in mice, but in the absence of any sort of obvious sign of disease. And I think this starts to raise questions about how adaptation to one particular cell type might actually come with trade-offs for pathogenicity of the organism as a whole. And so to wrap up this part of the talk, what I have told you so far is that we can use serial passaging approaches to select for Cryptococcus populations that have enhanced survival within phagocytes from diverse species, from amoeba all the way to mammalian or mouse macrophages. We can also use these approaches to identify particular genes and mutations within those genes that contribute to adaptive phenotypes. So I told you about the CAC1 mutation that we uncovered in this macrophage adapted strain that contributes to enhanced growth in macrophages. We can also use this to watch the evolutionary process. So we saw that this CAC1 mutation rose early and swept quickly through the population, which tells us something about the fitness effects of this particular mutation. And then I close by telling you about this really intriguing piece of data where this strain is able to establish persistent infection in animals, but does not seem to cause an obvious disease. And so I think, again, this raises some interesting questions about how adaptation to particular scenarios might impact the organism in other settings as well. And so there's still a lot of open questions on this story. So despite the fact that we know a lot about what's happening downstream of this CAC1 mutation, we still don't actually know how that missense mutation actually impacts the function of the protein or its interactions with other partners in the cell. And so this is still really an active area that my lab is working on now. We're also really excited about our ability to now use this evolved strain to probe differences in host responses. So how do these host responses look different in animals infected with our parental versus our evolved strains? And can that teach us something about persistent infections versus ones that are caused by more acutely pathogenic strains? And then, of course, we have all of those other populations. So we're really excited about trying to understand how this evolution looks different when you're adapting to an amoeba, or if you're this M3 strain over here and are using an entirely different set of mutations to adapt to macrophages. So I can tell you there's no overlap in the mutations that we've recovered between M3 and M1. And so this is work that has been picked up by a grad student in my lab, Jake Lanus, and I'm really excited about some of the stuff that he is already finding. So stay tuned for more of that in the future. But in my remaining time, I just want to pivot a little bit and tell you about a brand new story that we just put up on bioRxiv, which is work that was really spearheaded by a technician that I worked with while I was a postdoc, Mara Schwieso. And Mara's one true love is studying the impact of climate change on biological systems. And I convinced her for a very short period of time that she should apply this to the study of fungi instead. And so what Mara was really interested in was trying to build a system where we could use experimental evolution to look at the impact of rising temperatures on fungal evolution. So we all know that our climate is changing, but we don't have a great understanding of what this actually means for fungal evolution and further what this means for the emergence of new or deadlier types of fungal pathogens. But we're starting to get some hints about the impact of climate on fungal evolution by looking at simple epidemiological data. So here I'm showing you a very US-centric, view of some of the major endemic fungi. So we're looking here at histoplasma, coccidioides, and blastomyces. And when these fungi were sort of initially recognized as endemic, this was their geographic distribution. So you could see they really were, at least the sampling that we had, suggested that they were restricted to only certain parts of the United States. Over the next couple of decades, however, what you can see is that the case incidences of infections caused by these species has expanded dramatically across the United States, to the point where I'm not actually sure that we can necessarily call these endemic so much anymore, because they really affect the entire country. And so I think this is really quite striking, and there are many reasons that could explain the dramatic increase and the shift in habitat or geographic reach of these fungi. One is that the permissive habitats are expanding. Another is that these fungi are actually adapting to the changing world around them. I think the answer probably lies somewhere in the middle of these, but we're of course really interested in trying to understand how these fungi could actually adapt to changing environmental conditions. And so Mauro is really focused on trying to understand how these rising environmental temperatures influence fungal evolution, and more specifically, trying to pinpoint how thermotolerance evolves in fungi. And we think of this as really important because we often think of a primary restriction factor for fungal infection as being human body temperature. So many fungi cannot grow at this species, and that restricts them from being can't grow at that temperature, and that restricts them from being pathogens. But how is this going to change as the temperatures they encounter out in the environment actually increase? And so the first thing that we wanted to do was to actually use our favorite fungus, so Cryptococcus EvoFunPath, to see how much variation there is in thermotolerance across strains of this species. I can tell you, I'm not showing you the data here today, but when we look at clinical isolates, we see that they are almost uniformly able to grow at high temperatures, but it's a little bit of a different picture if we look at some of these environmental isolates. So all of the ones that I'm showing you here on this slide come from environmental sources, with the exception of our control H99 up here. And so what we see when we look at just simple spot dilution assays on a rich media is that these environmental isolates are all able to grow quite well at normal laboratory temperature of 30 degrees. But if we shift them up to 37 degrees, which is representative of human body temperature, we start to see some differences come out. And so there are some environmental strains that are able to grow very well at this high temperature. So FTC2171 shown here is growing quite well at this heightened temperature. Others grow less well, and the most pronounced example that we've found so far is this strain FTC239-1, which is actually the closest relative of FTC2171. These names are a handful, sorry, but is totally unable to grow at 37 degrees, or really has this very attenuated growth at these high temperatures. And so we saw this strain as an opportunity to investigate these questions of how thermotolerance might evolve in this species. And so what Mara did was she took this FTC239-1 strain and she serially passaged it while gradually ramping up the temperature over time. She started this experiment at a temperature of 30 degrees and ended it at a temperature of 35 degrees Celsius, so not going all the way to 37. This allows us both to keep some level of separation from human body temperature, but also lets us assess questions about whether adaptation occurs to a specific temperature set point or whether this happens to high temperatures more generally. And she did this experiment with 8 total replicate populations. These were evolved in 2 different media types. I'm not going to go too much into the details, but I'm happy to talk about it later. And at the end of this, she isolated both the populations, but also a number of single colonies from each population, which she then profiled for growth at her endpoint temperatures. And so I'm just going to show you 3 of the colonies that she focused on from 2 different evolved populations. So those are shown down here. So this colony here, which we call TA11 from our TA or thermally adapted population 1. What we see is that at 30 degrees, it looks very similar to our parental strain shown up here, but at 35, it is now much better able to grow. That the same is true for these 2 other colonies that Mara looked at down here, TA82 and TA83, both coming from the same population. Again, we see this enhanced growth at high temperature. And really kind of interestingly, if we also look at these strains at the higher temperature of 37, we see that they have enhanced growth at temperatures even beyond the bounds of what was used during this experimental evolution. And so I think this is really fascinating and teaching us something about how thermal tolerance might confer kind of general advantages across all high temperatures, not just at the sort of endpoints here. I should also say that Mara tested a total of 36 single colonies from the 8 populations that she evolved. And the vast majority of these did evolve some amount of thermal adaptation, so do grow better at these high temperatures than what she started with. And so this seems to evolve quite readily and actually quite quickly in this species. So this is pretty exciting, but Mara was not yet ready to declare victory. Instead, she found these assays, these spot dilution assays, to be a little static, and she wanted to know whether these evolved strains are actually able to outcompete the parental strain in a scenario that's much more similar to the setting that they are evolved in. And so what she did was she developed a parental strain that was labeled with a hygromycin resistance cassette. This allows us to differentiate it from test strains which don't carry this cassette. And so she can then mix one-to-one cultures of the parental strain with a test strain of interest, grow them at different temperatures for defined periods of time, and then measure how many cells come from each population at the end in order to determine a competitive advantage. And so what do the results of these sorts of experiments look like? So we'll start first with an important control. So the first thing that she did was she took an unlabeled version of the parental strain and competed it against itself. On the y-axis here, we plotted the competitive index. So you can think of this just as the number of strains coming, or the number of cells coming from the test strain versus the labeled parental. And here we see a competitive index of 1, which indicates that these two different strains are growing equally, both at 30 degrees and 35 degrees, which is exactly what we expected to see, and also tells us that the introduction of this drug resistance cassette does not cause any sort of fitness advantage or disadvantage under these conditions. So with this important control under her belt, Mara then took one of her thermally adapted isolates, this TA11 isolate, and competed it against the parental strain. When she does this, she sees that the strain is now much better able to outcompete the parental, both at 35 degrees, where we see this really high competitive index. So we recover many more cells from the TA11 strain than from the parental. But this is also true at the lower temperature of 30 degrees. So it seems like these strains are just better able to grow across many different temperatures. The same is also true for the other colonies that Mara looked at, although to a lesser extent. So we see high competitive index at 35 degrees, and then still some amount of increased competitive index at 30 degrees, although much less than for that previous strain. So in addition to this, Mara has also spent a lot of time trying to classify lots of different phenotypes that she's observed in these thermally adapted strains. We see a lot of mucoidean strains that are better able to grow at high temperature, which is pretty fascinating. There's also a number of morphological differences that she's analyzed, which you can see in her preprint that's now on bioRxiv. But I'll just highlight one of the phenotyping experiments that she did, which was to ask whether these thermally adapted strains are better able to resist other sorts of stress. And she focused in particular on antifungal stress, which is a more clinically relevant one. And so Mara did a very simple experiment just looking at the MICs of her different strains for fluconazole. And so you're seeing e-test assays here. I know they might be a little small and hard to see, so I've indicated our reading of the MIC in the bottom left corner here. And so when she looks at her FTC239-1 parental strain, she observed that the strain has an MIC of 8 micrograms per mil for fluconazole. Interestingly, in one of the thermally adapted strains, the MIC goes up. So we see that the strain is more resistant to fluconazole. And I should say that she's also replicated this in other formats as well. So this seems to be consistent. But we also see the reverse effect in some of our other thermally adapted colonies. So in both of the colonies that were derived from the TA8 population, we see that they have a reduced MIC and are more sensitive to this fluconazole insult. And so I think this is really pretty intriguing and also a little bit terrifying to think that adaptation to particular stresses such as increased temperature might not only change the number and pathogenicity of strains that we're seeing, but also may have impacts on our ability to treat infections caused by these strains. Okay, and so then I will close this out by telling you just a little tiny bit about what we know about the genetics that are underlying some of these phenotypes. And so this is work that has really been picked up by Marina Ruzic, who's a grad student in my lab. And when Marina started, I gave her this whole list of mutations that we had found by whole genome sequencing the 2 populations that I've focused on so far. So here you're seeing all of the fixed mutations that are contained within these populations. Everything is even messier than what I'm showing you here. So within individual colonies, we also see additional mutations. There's a lot going on here. But there are a couple of things that stood out that we kind of immediately wanted to test. So one of the things that we've noticed is that there are recurrent mutations in certain pathways. So in both of these populations, we've recovered what we think are likely loss of function mutations in Hog pathway components, so in 2 different kinases within the Hog pathway. The Hog pathway is involved in cellular stress response, and so this seems to make some amount of sense and seems like a prime candidate for driving some of this thermal adaptation that we're seeing. I'll also highlight one of our other kind of interesting candidates here, which is this missense mutation in a hexose transporter called HXT1. This hexose transporter has been implicated in transport of hexose sugars, but also in infection in animal models. And so we're particularly interested in probing this. We're still in the midst of doing the actual allele swaps for these different mutations that you're seeing here, but Marina did the quick and dirty experiment of just knocking out these genes in the FTC239-1 genetic background to see if complete loss of function might have an impact on thermal adaptation. And so these are super new results, so take this all with a little bit of a grain of salt, but just to remind you of the phenotypes that we started with. So here's our parental. It has this growth defect at 37. Our evolved colony TA82 is better able to grow at 37. And when Marina knocks out that SSK2 kinase from the Hog pathway, what she sees in 2 independent CRISPR knockouts of this gene is that the strain looks remarkably similar to our starting parental strain. So it doesn't seem like loss of SSK2 actually contributes to any sort of increased growth at 37. In contrast, when Marina instead knocks out that hexose transporter HXT1, my slide is very jumpy, sorry. What she sees pretty excitingly is that cells that carry the loss of this hexose transporter look much more similar to our evolved TA82 colony that has this thermal adaptation phenotype. And so we're starting to think that even though we were really intrigued by these Hog pathway mutants, it might actually be the unexpected HXT1 that's driving the phenotype that we're seeing, at least in some of these strains. And so with that, just to wrap up, I told you how we can also use experimental evolution to study thermal adaptation. And we see that this process happens quite readily and very quickly in this Cryptococcus neoformans strain. This thermal adaptation is accompanied by changes in cell morphology, but also resistance to other sorts of stresses, including the clinically relevant antifungal stress. And there are a lot of mutations that arose in our thermally adapted isolates that we're still kind of working our way through, including some in known stress pathways. But at least so far, the most promising candidates are not necessarily the ones that we expected. And so we're really excited about the new biology that we might be able to learn about thermal adaptation by continuing to study some of these mutants. And so then just to zoom back out, hopefully I've convinced you that experimental evolution provides a really unique and new view of these evolutionary processes in pathogenic fungi. And I'm really excited about how we're continuing to apply these sorts of approaches to ask questions about how all sorts of different environmental insults can influence the evolution of these species, which I think is going to reveal some really exciting new biology with important implications for human health. And so with that, I'll thank everybody who made this work possible. So my lab is now up to 5 people, which is really exciting. So this is all 5 of them trying to solve the problem of how to pack pipette tip boxes into recycling bins. Let's just say we're biologists and not engineers. It did not end well, but they're a great crew. You saw some work from Marina, who's in the middle here. The thermal adaptation story was started by Mara Shriso, who is a technician who worked with me in Utah, who's now a PhD student at UC Merced. All of our Cryptococcus strains were sent to us by John Perfect's lab at Duke, and we got a lot of help from Jenny and Dina in his lab. The mouse experiments and a lot of the characterization of cell size for the host story that I told you was carried out in collaboration with Jessica Brown's lab at Utah. And I need to give a special shout out to Jo in her lab, who really did a lot of work towards that project. I'll thank funding sources, and then I am happy to take any questions.
All right, wonderful. Thank you so much. That was fabulous. Very cool. Well, before we break off to kind of student only time, we can open this up to kind of general broad questions. So students are welcome to ask questions, though can save them for later. If anyone else has any questions, feel free to chime in. Yeah, wait, was that a hand?
Yes.
Yeah.
I really appreciate this this seminar. Thank you so much, Zoe, for such an overview on this idea. And my question related to the first part of your talk, when you put the fungi in contact with macrophages, what are the inflammatory responses, if there is, of the macrophages? You know, they are always producing certain molecules to protect themselves and so on.
Yeah, it's a great question. And I'll admit that the characterization of the host response is the part of the story that is still really missing. So both in vitro, but also in vivo. We've done a little bit of looking at cytokine production in response to infection with these different strains. Those assays so far have been a little bit noisy, so I can't really say for sure. But I think there might be some differences there that could explain some of what's going on. And then in vivo, we've also looked, we've done a lot of work to try to get at this, including some histopathology, and we don't see very pronounced differences in inflammation between the lungs of animals that are infected with the parental strain versus our evolved strain, which is kind of what we had expected to see. So I think there might be something more subtle going on, but we have not yet been able to get to what that is. But it's a great question.
Thank you so much.
Yeah.
Great. Other questions? Matt.
So can you estimate like how many sort of cell divisions happened in some of those intracellular, directed evolution experiments?
Yeah. So you're asking about generation time, I'm guessing, right? So it changes over the course of the experiment. And I think one of the really fascinating things, especially with the M1 strain that I talked about, is you can actually tell there are morphological differences, but also the speed of replication within the macrophage is so different. So in some of our mixed population experiments where we had, from like the middle of our experiment, we thought out passages where like 60% came from M1 and 40% from the parental. You can actually tell which macrophages got which genotype, which is pretty cool. But in terms of the number of generations, we think it was somewhere between 50 to 100 in the entire experiment. So really not that many. And I think to get to passage 4, significantly less. So probably on the order of like 20 to 30 generations before we see this mutation arise.
Other questions? Nick.
Hi, Zoe. Thanks. That was a great talk. We're evolving some Candida pathogens in heat, and we find that we get a fair amount of bacterial contamination as we're passaging. Do you ever see that? And do you have any advice on what you can do to avoid that?
Did you review our paper? Yes, we did also see bacterial contamination. I think we've actually, we didn't totally realize that it was there until after we had sequenced. So we now have like a high quality assembly of this bacterial contaminant, which I'm pretty sure was derived from Mara's skin microbiome. So it's what we're seeing is mostly staph hominis strains, which we think are coming from us. It's a hard question. We've thought a little bit about doing this in the presence of antibiotics, but I think then you might end up kind of co-evolving strains that are antibiotic resistant, which is not what we're going for. I think the best strategy we've come up with is just to do everything in as sterile an environment as we can. So we've switched over to doing all of these experiments in a biosafety cabinet, trying to minimize the amount of kind of outside exposure to other microbes as we can. But yes, we absolutely have had that problem. We don't think it actually impacted the evolution in a serious way, although it definitely impacted some of the metrics that we used over the course of our evolution, for sure. But interesting to hear that other people have this issue too.
I think I saw one more hand by one of the Elisa Gersteins, but I don't think it's necessarily Elisa Gerstein.
Hi, can you hear me?
Yeah.
Oh, I'm Aruni from Elisa Gerstein Lab. I have a question about your competitive assays. So when you get the competitive index, is that the difference between Malthusian parameters of your competitors? Is that how you calculate that one?
Yeah, so we calculate, yeah.
What I'm asking is that W or R, like is it a ratio or like subtraction between them?
So it's a ratio. So you can essentially, if the test strain is out competing the parental by a factor of 2, then your competitive index would be 2. So it scales according to that. All of the details are in the paper, but I'm also happy to talk more about how we do this at any point. But yeah, it's a ratio of the number of cells coming from each population.
Oh, okay. And like how many replicates did you use, like bioreplicates, to get the final count?
Those experiments can be quite noisy, and I think much to Mara's chagrin, the data that you're seeing there was like, I think it was a lot of time to get it optimized, and so she did 3 biological replicates in at least 3 different experiments. So what you're seeing there was the average of biological replicates from each experiment that she did.
Okay. I have this, because I am doing a competitive fitness assay too, and it is like kind of different than this one, but the idea is the same. So I just want to make sure that my replicates are in the like acceptable noise range. So when you say you did like 3 replicates, can you give me like a general number, like to get that one, like in the initial competitive assay, to get that one, how was the numbers between the replicates, like before averaging them?
Yeah, I think this is definitely
all over the place.
There was a period of optimization, partially because we were working really hard to get the cell numbers correct, because partially, so there are differences in cell size between the adapted strains that we recovered and the parental strain, and so to get an actual one-to-one mix was sort of a challenge. We do see, I think by the end, the noise had definitely decreased quite a bit, and so we saw a fairly tight correlation between biological replicates in a given experiment, and that's when we felt pretty good that things were working. So I think there has been a range of variation in any given experiment, but I think for us also running the control in every experiment and seeing that we're not that we're getting sort of a competitive index of 1 for that all the time also made us feel better about the results, but they definitely can be a little bit noisy for sure.
Okay. So one more question. So if you end up getting your average at 1, let's say in the first part, you think that all the replicates are in the acceptable range. Is that it? Like,
I'm not sure I would say that's necessarily true. So I can also tell you that she initially made a strain with a gnat resistance cassette,
Okay.
and we saw very different results with that. So we saw that
Mhm.
strain actually had a fitness defect consistently across experiments. And so
Okay.
she had to remake her control strain that we were using as the parental competitor, and
Mhm.
had to kind of figure out which of those did not have a fitness effect when we integrated this drug cassette. So I'm not sure that just seeing a value of 1 necessarily means that the experiment is usable, but I think that is an essential control to have in place before you can really consider your other test strain
Okay.
data. Yeah.
Oh, okay. Thank you so much. It was a great talk. Thank you.
Thank you.
All right, wonderful. Well, I think since it's 12, we'll draw an end to the seminar. Join me again in thanking Zoe for a fabulous talk. Thank you. And now we will, non-fellows will log out. I will hand this over to Dominica and yeah, take it from there. Awesome. So thanks so much, Zoe.
Yeah, thank you.
Thank you so much, Zoe, for your amazing talk. So can you guys hear me okay?
Yeah.
Okay, perfect. So my name is Dominica. I'm the discussion lead for today's session. And so these sessions are usually quite informal. It's just an opportunity for the fellows to sort of ask you about your research, but also about sort of your career trajectory and sort of things like that. I will just ask if any of the fellows are able to turn on their cameras, if you have the appropriate bandwidth, that would be awesome. But with that, I'll sort of open up the floor for questions. Would anyone like to start? Okay,
Yeah.
perfect, Brianna.
Hi, Zoe. That was a great talk. I also study Cryptococcus, so I always love hearing other Cryptococcus researchers. I'm wondering, for the macrophage experiments, did you increase the survival time for the mouse assays to see if the M1, even though they were persistent, if the mice eventually showed any symptoms?
Yeah, that's a great question. We have not done that. Part of this was because all of those experiments were done in collaboration. I didn't want to ask them to do the 6-month experiment to see how long it would actually take. So that's one of the things. We're still getting our mouse protocol up and running here at BC, but that's definitely one of the things on our to-do list, is to set up those infections again and follow them out to, I don't know, we'll probably set some arbitrary endpoint in case the animals really never actually develop disease. But I suspect they will at some point, but I'm not sure when that will happen. The other thing I can say that we did do is we tried, so Jessica's lab had previously seen some of these isolates that don't seem to cause as much disease as others, and they had experience with immune suppressing those during the course of infection, and then seeing that the animals would actually go down rapidly after immune suppression. So we tried to do those experiments, and the animals were totally fine. So even with, I think it was 2 weeks of immune suppression, we didn't lose any of the animals.
Interesting. Did you still see like fungal burden associated with the lungs and brain following immune suppression?
We did, yeah, and it went up. So we saw more fungus in both the lungs and the brains of the animals that experienced the immune suppression, but they were still fine, at least as far as our assessments of them. So, yeah.
Interesting. And did you see any virulence trade-offs with like the point mutation, like capsule melanin, anything like that?
Yeah. Yeah, so that's all the data that I didn't talk about today, partially because it just takes a little longer to explain all of that. And I wanted to squeeze 2 stories in. But yes, we see, so in the strain that carries the point mutation, we see decreased capsule size. We don't see decreased melanization at low temperatures, but we do at high temperatures. And then I think probably the biggest difference that we see is that the cells that carry the mutation are unable to form Titan cells, so they don't get big. And we've seen that both in vitro, but also in vivo. So if we take the lungs out of the mice, we see that the animals infected with the parental strain have really big cells in the lungs. But if we look at the same the same time point for animals infected with the M1 strain, we see cells that are uniformly much, much smaller. And so we think that might be part of what's going on, is that they're not able to get big.
Right.
But yeah, there's a lot of changes downstream. It's I think it's a little hard to know what's the most important, but
Yeah.
yes, it definitely impacts those phenotypes for sure.
Interesting. So they have like a better infinity for the macrophages, but that doesn't translate to virulence in vivo. So do you think like they're still surviving within the macrophages and like that allows the persistence?
Yeah, we tried to look at this with some of the histopathology to see if we saw more of an association with macrophages in the lungs. We couldn't see anything significant. I think the real experiment would to actually be to try to like get macrophages out and look at by, I don't know, flow or something to see if there's more of an association with the M1 strain and the macrophages in vivo. But we don't have that data. So not totally sure. We did find some cells in macrophages, but that was true for both strains. So
interesting. Those are my questions for now. If anyone else has any questions, thank you.
Thanks, Brianna, so much. So I just sort of have a question. It's sort of maybe more general, but your research right now is focusing a lot on Cryptococcus neoformans, and it sounds like your lab is newly established. I was just wondering, do you plan on expanding beyond Cryptococcus neoformans for now, or what's sort of your next stage if you were to pick a different fungal pathogen?
Yeah, that's a great question. I think for most of our experimental evolution stuff, we'll probably stick with crypto for right now. That's just partially it's what I know the best and am best able to help advise people in my lab with. But what I didn't talk about today is another big part of my postdoc research was actually looking at host evolution. And so looking particularly at receptors in the host immune system that engage with different types of fungal pathogens and how those are evolving across mammals. And so there's some really interesting stuff there. And I think for that will almost certainly take us into the space of other types of pathogens. So for instance, there's some kind of interesting evolution happening in rodent dectin-1 and also in bat dectin-1. So we see different sequences of those receptors. And I think dectin-1 is not a receptor for Cryptococcus. It does not seem to recognize Cryptococcus. And so that would necessarily mean moving into like Candida space and trying to understand how differences in these immune receptors from different species might recognize Candida differently. So yeah, I'm definitely interested in other fungi. I don't know that I have a favorite at this point, but I'm sort of one of these people who kind of follow the science wherever it leads me. And so with the immune receptor stuff, I think that's going to be really interesting to see where we go.
That does sound really interesting. See, I research Candida, so I was just sort of seeing if there was a pathway to that in the future, but I'll definitely have to keep following your work. Do any other fellows have a question? Because I have another one I can ask. Okay, so I'll go ahead. I'm really interested just to sort of hear a little bit about your career trajectory. So again, congratulations on starting your lab this past January. I'm sort of a senior PhD student, and so I'm sort of like looking towards my next steps, and I'm wanting to do a postdoc, but it sort of sounds like for yourself, you transition to pathogenic fungi through your postdoc from doing something different. So I was just wondering, like, if you could sort of talk a bit about like your experience and like what sort of drove you to change into a different area of research and sort of how you got to your lab now, because you're like very early career researcher. So again, congratulations.
Yeah. Yeah, I think my 6-month anniversary of being a PI is next month. So very, very baby at this point. But yeah, I think my career has definitely not gone the way that I expected at any point. So I guess to start from my undergrad, I was totally obsessed with protein biochemistry as an undergrad. I thought it was just like the coolest thing. I wanted to do structural biology or biophysics. And I was like, this is the dream. This is what I'm going to do. And so I went to grad school thinking that that was what I was going to do and mostly was interested in labs doing that. But I'd never taken genetics. And so I took a genetics class. I was actually pretty bad at it, but I really liked it. And so then I ended up joining the C. elegans lab and just like totally falling in love with worms and with genetics. And yeah, I still love worms. I would love to do worm stuff. But I really loved genetics and thinking about genetics questions and as a PhD student. And one of the things that I started off working on during my PhD was actually thinking about evolution of gene expression patterns. And so I was particularly interested in worms. We know that they can be infected with all sorts of bacteria. We know a little bit less about fungi and worms, but we had discovered this gene that turns on when you infect the animals with a certain type of pathogenic bacteria. And so I was really interested in looking across many different worm species to see if that gene expression was different if you looked across these different species or different strains. And that was kind of what I started off my PhD being interested in. That project was much slower to progress. And so I ended up doing something else, which is probably something that's familiar to some of you, as my guess. And so when I reached the end of my PhD, what I really wanted to do was to get back to that idea of kind of natural variation and evolution in those host pathogen interactions. And so it seems like a big switch, but it was actually kind of going back to something that I had been interested in from the very beginning. And so when I picked my postdoc lab, the lab as a whole is kind of interested in the evolution of host pathogen interactions. And so most of the lab actually studies viruses. There were a couple of people at the time that I joined that worked on bacteria, and then I showed up and my postdoc mentor was like, what if we try fungi? And so it was not, I would not say it was like a calculated move. I had no idea that that's what I was going to end up doing. But I started reading and I fell in love with the idea of fungi. And then I started going to fungal pathogens meetings and just really loved the community. And so that has really kind of kept me in it. But yeah, it seems like there was a total 180, but there was a thread there. But it was still kind of a very new thing for me, especially adding in the fungi and doing that in a lab that wasn't really set up to do any of that. And so I leaned pretty heavily on collaborators like Jessica's lab at Utah.
So your postdoc, you said, was at in Salt Lake City, Utah?
Yep, that's right.
So you've done like lots of moving around too then for
yeah. Yeah.
Well, that all sounds like really, really cool. So I see that you had sort of like this thread that sort of brought you back, which is like really interesting to hear. Yeah, and I myself, in my PhD, you know, it's always a bit of a mess. So I feel like it's a shared experience of what kind of goes on with like the changes and different directions we all kind of go down.
Yeah, for sure.
But I always just like hearing it from, you know, someone who's very much so senior to me and is in like a really great position. So for your lab right now, you said you had like 5 lab members. Are they mostly like grad students that you've sort of hired on, or?
Yeah, so currently we have 2 grad students who actually started in the lab. They started their rotations in the lab the same day that I started my position. So they have been in the lab since day 1, and then they both joined, which was really lucky for me. And then we have a lab technician who just joined, I guess about a month ago now. And we have 2 undergrads. So they are just kind of learning science. Neither of them has ever worked in a lab before. And so they bring a really fresh energy, but also a little bit of chaos. So it's been fun, but we're also interviewing postdocs right now. So I think eventually I like mentoring students, I think, a lot. And so I envision my lab will always be a little bit student heavy, both grad students and undergrads, but I think postdocs bring kind of a new perspective and a different set of skills, which is really useful. So I think we'll probably also have a few postdocs around too, but not anticipating having like a giant lab group at any point. I'll say even having 5 in the lab currently already feels like a lot. So.
Well, that's like all very exciting. I'll have to keep note. You had said, I'm gonna like a lot of what you presented today, that those results have been published, right? Or just the first part has a thermotolerance? Oh yeah, you said it was on bioarchive. Okay, I'll have to like, look them up just to reread and like summarize everything. But does anyone else have any questions that they'd like to ask Zoe?
I have one more question. Was going the PI route always like your goal? You know, did you always, when you started your PhD, you were like, professorship, let's go? Or, you know, was there?
No.
Yeah.
No. Yeah, no, I mean, I still have days where I'm like, is this what I'm doing? Is this what I want to be doing?
Yeah.
I don't feel like I've ever actually known that this was like 100% the job that I wanted. I don't know if that's helpful or unhelpful to hear, but I can only be honest about that. I think the things that have kept me in it is my research interests have always been really fundamental biology. And so I think there probably are other jobs that I would find really exciting and interesting in other ways, but at the end of the day, the thing that keeps me up at night is like really basic biological questions about whatever it is that I'm working on, whether that's worms or fungi or amoeba, right? And I like being able to study those things. And so it's a little bit harder for me to imagine those passions finding a home in other sorts of positions, particularly industry. So that's not to say I wouldn't be happy there, but I think that's really been the thing that has kept me in academia, is being able to study these weird things that I find so fascinating. But
yeah, No, it's very relatable for sure. Because I'm also close. I'm a senior PhD student, so I'm about to defend. But yeah, at night I'm like, which way do I go now? Like, should I go for a postdoc, or should I pursue other fields? But I'm very similar. I love fundamental biology, but it's a more difficult career choice, with going the PI route and everything. So did you always what interested you in Boston College? Like, there was a opening and you applied. Did you apply to a lot of other universities? If you don't mind me, like the process.
Yeah, totally. So I applied really broadly across the United States. I also applied to some positions in Canada, but never heard back from any of them. But I applied kind of all over the place. I do have a partner who's also a biologist who's in biotech. And so we didn't restrict my search based off of that. But when I ended up with options in a place like Boston that has a lot of options for him, that made it a little bit easier. But one of the things that I really liked about this department and why I ended up here is that it is a little bit of a smaller community. So we're not a huge department. And we're kind of specialized in two areas of biology, but we're not like a microbiology department, we're a biology department. And so there are a bunch of people who do microbiology, and then there are a bunch of people who do kind of cell and developmental biology. And so I liked that there was a breadth of different questions and systems. And so I'm the only fungal person, but there's a toxoplasma lab down the hall. So there's another eukaryotic pathogens person. There's a couple of bacterial people. There are a bunch of people thinking about the microbiome. And so I'm trying to convince them that they should be thinking about the fungal microbiome too. But then I can also go talk to my colleague who studies like cell division in Schizosaccharomyces pombe, and it's like very different, but shares a different kind of language with me. And so I liked that there were all of these intersections, but there's also just a lot of different types of biology going on. I think that's kind of where I thrive the most. And then I really enjoy teaching. And so I was actually specifically looking for positions where I would have undergrad teaching responsibilities. And so I have those here. They're not like I teach a class and a half a year, which is not that bad. But I enjoy it. And so that was something that I was looking for as well.
Nice. Well, it sounds like a really nice community, like going on at the department. Because like big universities, it can be noisy or a lot of.
Yeah, I think there can be like really big departments, and maybe I would thrive there too. I don't know. And then there can also be, I interviewed in a number of very specialized departments, right? So like microbiology and immunology departments where like everybody was very, very similar to what I was doing. And I think there's appeal to that as well. There's a lot more people around who like have the equipment that you need and who can read your grants and give you really detailed comments on them and stuff like that. But I think for me, those people can still be my colleagues. I'll still see them at meetings and all of that. And I like being in a department where the person next door thinks about things in a very different way from what I do.
Yeah, absolutely. No, that's such a good perspective. That's great. Well, congratulations, by the way. I don't think I've said that. Yeah. I don't have any more questions as of right now.
Okay, I'll do a last call for questions from the fellows, and then we can wrap this session up. Okay, so with that, again, thank you, Zoe, so much for taking your time, for sharing your research and also your sort of trajectory with us. I think that you gave us a lot of valuable information that we'll take with us throughout our studies. And I personally look forward to following your lab, so I'm definitely going to go check out your papers and read a little bit more into your research. So
yeah,
thank you.
Feel free to reach out. I'm happy to talk to anybody about our work or life or science or whatever at any point. So don't hesitate to reach out.
That is wonderful.
I do
go
have
ahead,
one more
Brianna.
question. Sorry.
For sure.
Have you ever thought of doing like evolutionary experiments in vivo?
I thought a little bit about this. I think it would take the right person to come along and really want to do that because it's a little bit of more of a commitment,
For sure.
I think. But yeah, I think there's some really interesting, especially there's some cool Candida stuff where they've passaged Candida through mice where it's not normally a commensal, and then they find it changes in ways that it becomes more like a commensal. So there's some really cool stuff with that that I've always been a little bit jealous of. But I'm not ruling it out. I think it would just take somebody coming along who's really invested in that for us to pick it up.
Yeah. I was just thinking that'd be cool to see the changes.
Yeah, for sure.
Okay, that's my question. Thank you so much.
Okay.
Yeah, thank you.
you.
Okay, have a good day.
All right,
Bye.
bye.